Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this gluconeogenesis lecture, I will be explaining you on regulation of gluconeogenesis. Let's get into the details. Now as you know that gluconeogenesis is the pathway that runs under fasting condition and starvation. The importance of this pathway is to maintain a blood glucose level under fasting condition so that the tissues like brain and the cells like red blood cells, they get a needed glucose for their energy purpose. So since the reactions in gluconeogenesis are kind of antagonistic to reactions that are seen in glycolysis, so these two pathways are antagonistic. So because the glycolysis is a breakdown of glucose into pyruvate, whereas gluconeogenesis is the formation of glucose from pyruvate, so they run in opposite direction. That's why gluconeogenesis and glycolysis, they are antagonistic pathways, and that's why they both must be tightly regulated. Okay, so what type of regulation we have here? So the regulation on gluconeogenesis can depend on substrate availability, allosteric regulation, hormonal regulation, and regulation at gene level. So we are going to study regulation of gluconeogenesis in 4 separate groups, that is substrate availability, allosteric regulation, hormonal regulation, and regulation at the gene level. Now let's get into regulation based on substrate availability. So whenever person goes into fasting condition, at that time the skeletal muscle proteolysis which will be going on because of low insulin and higher cortisol level, and this happens especially when the person is in stressful situation and fasting condition. So low insulin, increased cortisol, these are the two things that can lead to increased availability of gluconeogenic precursor, and that is alanine and other gluconeogenic amino acids. So substrate availability will lead to increase in gluconeogenesis process. Similarly, when a person is in fasting condition, hormone glucagon will be increased, and this glucagon, it is going to stimulate hormone sensitive lipase enzyme, thereby break adipose tissue triacyl glycerol into free fatty acids and glycerol, and the glycerol is a gluconeogenic substrate, so glycerol gets into gluconeogenesis. So that's the substrate availability there. Anaerobic glycolysis providing lactate, and that lactate goes into gluconeogenesis, and also high protein low carbohydrate diet which will keep insulin low and glucagon high. So low insulin and high glucagon will also stimulate gluconeogenesis because there will be higher amino acids available, and these amino acids are diverted into glucose formation. Like this, availability of substrates can stimulate gluconeogenesis process. Now coming to allosteric regulation. In the allosteric regulation, one of the most important enzyme that is regulated allosterically is pyruvate carboxylase enzyme. Now the pyruvate carboxylase enzyme is positively modulated by availability of acetyl-CoA. Why acetyl-CoA levels are high during fasting and starvation? It is because fatty acids that are coming from adipose tissue lipolysis, they will undergo beta oxidation to provide acetyl-CoA, and the excess acetyl-CoA levels, it has two effects. One is it will have a negative effect on pyruvate dehydrogenase enzyme complex, and it will have a positive effect on pyruvate carboxylase enzyme, thereby it is going to divert pyruvate into oxaloacetate rather than pyruvate into acetyl-CoA. So when pyruvate is diverted into oxaloacetate, oxaloacetate will go into gluconeogenesis formation, and that's how gluconeogenesis is stimulated by acetyl-CoA acting as a positive modulator on pyruvate carboxylase. At the same time, it will have a negative effect on pyruvate dehydrogenase enzyme complex. Now, the other allosteric and hormonal regulation that is going on over gluconeogenesis and also on glycolysis simultaneously is the concentration or the availability of molecule called fructose 2,6-bisphosphate. Now, the fructose 2,6-bisphosphate, it simultaneously regulates gluconeogenesis and glycolysis. What are the enzymes that are regulated by fructose 2,6-biphosphate concentration? Now, the fructose 2,6-biphosphate, it is going to regulate PFK1 and it is going to regulate fructose 1,6-bisphosphatase enzyme. PFK1 is the most regulated enzyme in glycolysis. Fructose 1,6-bisphosphatase is the regulated enzyme in gluconeogenesis. So, note that the most regulated enzyme in gluconeogenesis is pyruvate carboxylase, which we have seen in the previous slide. But if the regulation on glycolysis and gluconeogenesis has to go on simultaneously, that has to be done by using fructose 2,6-bisphosphate. Let's look into that. Whenever person has got low blood glucose level, like fasting condition, fasting leading to low blood glucose level, so glucagon rises, and that glucagon, it's going to increase protein kinase A enzyme activity. So this protein kinase A is going to phosphorylate an enzyme called PFK2, and this PFK2 enzyme, it's a bifunctional enzyme. It has got kinase subunit, it has got phosphatase subunit. So whenever this enzyme is phosphorylated, so kinase subunit will become inactive, phosphatase subunit becomes active. So when the phosphatase subunit becomes active, at that time there will be conversion of fructose 2,6-bisphosphate back into fructose 6-phosphate, and thereby fructose 6-phosphate can go into glucose 6-phosphate, and the glucose 6-phosphate goes into glucose. There will be increase in gluconeogenesis in the presence of glucagon. That's what happens under fasting condition. Now coming to the fed condition. Whenever person is in a fed condition, at that time it's all about insulin. So insulin is going to increase protein phosphatase enzyme activity, and this protein phosphatase enzyme activity, it is going to remove phosphate from PFK2 enzyme, and that PFK2 enzyme, now its kinase subunit becomes active, phosphatase subunit becomes inactive, thereby fructose 6-phosphate now will be converted into fructose 2,6-bisphosphate, and there will be increase in fructose 2,6-bisphosphate, keeping PFK1 active. Now what happens is, when it's all about the concentration of fructose 2,6-bisphosphate. So fasting keeps fructose 2,6-bisphosphate levels decreased. Because of this, PFK1 enzyme activity decreases, whereas fructose 1,6-bisphosphatase enzyme activity increases. That means glycolysis decreases, gluconeogenesis increases. At the opposite way, whenever person is in fed condition, at that time, so the levels of fructose 2,6-bisphosphate will increase in the presence of insulin, and that will lead to activation of PFK1 and inactivation of fructose 1,6-bisphosphatase. That means glycolysis increases, gluconeogenesis decreases. So like this, allosteric and hormonal control is there over glycolysis and gluconeogenesis simultaneously. Now let's look at induction and repression done on key gluconeogenic enzymes. Now let's get into regulation based on induction and repression of key gluconeogenic enzymes. So the hormones that will act on key gluconeogenic enzyme synthesis are glucagon, epinephrine and glucocorticoid. When the person is in fasting condition and starvation, you know there is increase in glucagon. When the person is in stressful situation, there is increase in epinephrine. And when the person is in stressful situation, infection, that kind of thing, so there will be increase in glucocorticoids. So glucagon, epinephrine and glucocorticoids, all of them, they are going to induce the genes that are coding for pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase and glucose 6-phosphatase. All these enzymes are genes for these enzymes are induced. So the levels or the concentration of all these enzymes will be increased, and that leads to increase in gluconeogenesis, and increase in gluconeogenesis will increase glucose levels, and that will bring up glucose levels in the blood, and also sometimes it will lead to hyperglycemia. Now under fed condition, what will happen? So under fed condition, insulin, which is going to induce key glycolytic enzymes and antagonize the action of glucagon, epinephrine, glucocorticoids, and effects of these hormones on gluconeogenic enzymes. So basically, insulin, it is going to reverse the action of all these gluconeogenic hormones like glucagon, epinephrine, glucocorticoids. So whatever these enzymes have been induced by these hormones, it will be reversed by insulin's action, and also insulin is going to induce and activate all the glycolytic enzymes, thereby glycolysis will be increased and gluconeogenesis will be decreased. So like this, gluconeogenesis pathway is regulated. So that's all about regulation of gluconeogenesis. I hope this video has helped you in understanding how gluconeogenesis and glycolysis are simultaneously regulated by insulin and then glucagon, growth hormone, glucocorticoids, and epinephrine on the other side.